Guys, my name is Avinash, and on behalf of Bionavit Technologies Private Limited, I would like to welcome all of you in this webinar, which we have titled as Into the Researchverse, where eminent scientists from across the country and from various reputed research institutes share their research insights about next generation sequencing and how it has helped them achieve success in their research paradigms. As part of the same series, we have with us Dr. Jyoti Singh. She's a DBT-Welcome Trust India Alliance Early Career Fellow at NCCS Pune and has done extensive work in understanding the role of small RNAs in host pathogen interactions. Bionavid has got the privilege of being a small part of her research journey, and we are quite thankful to her for agreeing to be a part of this webinar series and share her research experiences and insight about genomics and its impact on research with all of you here. So I'll be handing over the session to Dr. Jyoti. You'll be hearing next from her. Hello.
Hello, good afternoon, everyone. Am I audible?
Guys, if you can hear Dr. Jyoti properly, you can type a yes in the chat section. And also about the queries, while the session is going on, while ma'am is interacting with all of you, while she's presenting her topic, just drop in your queries in the chat section so that I can keep a track. I'll try to moderate and cover all the queries once ma'am's talk is over. Fine. So I'd request you to not to interrupt in between. Just drop in your queries. I'll keep a track, and then we'll discuss them once the presentation is over. Over to you, ma'am.
Okay, great. So hope I'm audible, right?
Yes, ma'am. Yes, ma'am. Audible. Yeah.
So first of all, thank you, Avinash, for the kind introduction. And I would also like to thank Rohit for giving me this opportunity to share my experience with all of you. And I also like to extend my thanks to the whole Bionavit team for providing me wonderful service and support from past 5 years or so. And I hear that you got the best team for data analysis, and you guys don't let any stone unturned to scientifically satisfy your client's demand. And with this note, I think I'm going to start my talk. And this is on application of small RNA sequencing. I'm a biologist, so I have designed my talk in a way that you will get to know the glimpse of the how biologists utilizes these next generation sequencing as a tool to address the research questions and the objectives. And most importantly, I will try to clarify some of the common confusions we have among the researchers in regard to which NGS platform we should use, what should be the biological replicates one should use when they are going for a small RNA profiling, or they have to check for a differentially expressed small RNA, and what kind of coverage one should require. So I will be, so my main motive is to explain and clear all these kind of doubts. So to starting with, this is the kind of outline for my talk. So as I mentioned you, so my main focus should be to give you a kind of a guidelines for selection of NGS, and then I'll talk about my research. Well, as we know, the central dogma of molecular biology describe in two steps. One is transcription, another one is the translation. So when DNA transcribed into RNA, and then the RNA translated into protein. But we also know that there are some RNA which doesn't translate into protein, and these are the non-coding RNA. And if we have to broadly divide these non-coding RNA, we have mainly for three groups. One is the protein machinery RNA, the other one is small non-coding RNA, and the third group is a long non-coding RNA. So if you talk about small non-coding RNA, you can see there are multiple species: small interfering RNA, microRNA, small nuclear RNA, and PV interacting RNA, or in short we call it the pi RNA. Well, when we talk about small RNA sequencing, the three most relevant and focused RNAs are pi RNA, siRNA, and microRNA. siRNA can be of exogenous origin or can be endogenous. When I say exogenous, that means when the virus enters, an RNA virus enters, and the viral replicates and it produces a double-stranded RNA, and which further processed into a small double-stranded RNA, which we call it siRNA duplex. Wherein endo-siRNA originates when there is the transposable element production, or if there is a reverse transcription or convergent transcription, I should say, and there is a chances that there is an internal double-stranded RNA molecule production. MicroRNA, I think we all are well versed with what is microRNA, and then comes the pi RNA. Pi RNA act as a defense mechanism against transposable element, and they are nowadays shown to express not restricted to express only in the germ-like but also in cells. Coming to the different technique we use to see the expression of these RNAs, both microRNA as well as normal mRNAs. So the most commonly used techniques are Northern blot, RNA protection assay, PCR, qRT-PCR, microarray, NGS, and if the limitation and the advantages, we will see that the next generation sequencing has many advantages over all the previously used technique. And the first and foremost important is the de novo sequencing. So unlike NGS, all previous methods were, the prerequisite for them was to know the sequence, right? And because based on that, they used to make the probes. But with the NGS, we can also do a kind of a de novo sequencing. We don't require any sequence knowledge. It gives definitely a high throughput data and with very high sensitivity. It also helps in discovery of various novel markers, novel small RNAs. You can also characterize microRNA isoforms. So microRNA isoforms generally differ in 1 or 2 nucleotide at the 5' end, which is very sensitive. It shows that this technique is really sensitive. And beside that, we can also take advantage of the sensitivity of small RNA sequencing for finding out the differential express genes as well as small RNA. Okay, so let me talk something about the evolution of the sequencing techniques. So, because as we know that these all technique, NGS and all, they are high throughput sequencing, but they have come, I think, not a decade. For that, there were many other sequencing techniques which we have been using, and one of the most popular one is the Sanger sequencing. But before Sanger, there was this Maxim Gilbert sequencing, and they were the pioneer in sequencing, followed by Sanger. And in the Sanger sequencing, as we know, and I'm quite sure in the MSc you must have studied, that we use di-deoxynucleotides, and then once we a dideoxynucleotide mixture and repeat this cycle, and then we have to run a gel wherein we have a different nucleotide in a different lanes. This is a tedious process. That's why it has been improvised into, I'll say, the second generation kind of a machinery, wherein instead of using a whole big gel, they have now restricted it to the capillary. So you can see over here, instead of running multiple lane, you can use a kind of a capillary here. And further, it was improvised and called as the Sanger Part 2, wherein instead of picking each and every colony by hand, they have automated this process and hence reduced down this time taken by Sanger sequencing. So one of the drawback I will say with Sanger sequencing was definitely it takes a lot of time. But if you have, still we are using Sanger sequencing, because if you have 2 or 3 transcripts, and especially when you have to sequence a clone, we still are doing Sanger sequencing, and because it is accurate, definitely it is accurate. But yeah, as I mentioned you before, it is time consuming. Advantages of NGS over Sanger sequencing, I have already mentioned that it doesn't need bacterial cloning, number one. It's cost effective, definitely, because we don't need that dideoxynucleotides in this. It sequencing by synthesis, and that's how it takes less time, cost effective, and you can do a massive parallel sequencing using next NGS. Now, if we compare the popular platform for NGS, we will see, first we see how the timeline for its invention. 2004, I think it had started, around 2004, Roche was the first which came into picture, and they started doing sequencing. And then Solexa, then then further SMS, then RN Torrent, then SMRT, then Oxford Nanopore. And these are all single cell sequencing machines. These are just a picture to show what are the most popular ones. These are the most popular platform as of now. And for small RNA, we generally use Illumina. And Illumina has basically 2 variants. So I'll talk about it a little later. So this is just to compare different features of all these popular NGS platform. You can see over here what are the disadvantages and advantages of these different platforms. So for high throughput data generation, we generally use HiSeq, and also it is cheap. And the only drawback it has, that since it has a short reach, so it takes long as compared to other platform. Yeah, so when we talk about Illumina and we talk about small RNA sequencing, so the most popular variants are MySeq and HiSeq. NextSeq and the other one is not that popular, especially I'm not aware of, but yeah, so I can talk about MySeq and HiSeq. So if you compare MySeq and HiSeq, you will get to know that if you need a lot of reads per run, you should go for HiSeq, because the number of reads per run is really high in HiSeq as compared to MySeq. But if you really want it quick, then HiSeq won't be a good choice, because it takes definitely more time, almost a double time that what MySeq take. But then HiSeq is definitely costly as compared to MySeq. So we'll, in the later slide, I'll try to find out a solution if you have a problem, and you're not unable to, and you're unable to actually come down to any solution that which variant of Illumina I have to select. So we will talk all these things later, maybe after few slides. One should go to which variant. Application of NGS, I think by now we all know that this is a very powerful tool. We can go for whole genome resequencing, we can do de novo genome sequencing, targeted sequencing, transcriptome sequencing, metagenomic sequences, and of course small RNA sequencing. So now this slide onward, I will focus only on the small RNA sequencing. To start with, what are the different steps involved in small RNA sequencing? Number one, and the most important step, is the experimental design. Number two is the RNA preparation. Number three is the construction of library. Number four is the sequencing. And finally, analysis of the data, whatever we get it from sequencing. So when we talk about experimental design, there where we as a researcher come into picture, because it is very important to design your experiment beforehand. When I say design your experiment, I mean that you should know why you are going for this NGS. And you should also know what kind of expectation you have from the outcome of these NGS, right? So first decide what you want to do. Why you have selected NGS? Can't this kind of objective being addressed by any other technique? So you should be very clear that why do you want to go for NGS. Second comes RNA preparation. Definitely RNA is, as we know, that it's not a very stable molecule. So when you isolate RNA from either tissue or cell, be careful. Okay, so generally we isolate total RNA and then we subject it for the construction of a library, because then there are multiple kits, commercial kits available, which will take care of the size. And construction of library basically consists of adapter ligation step, and then conversion of RNA to cDNA, and then followed by a sequencing, because it is sequencing while synthesis. So sequencing and synthesis of cDNA complementary strand will be together in parallel, and finally a bioinformatic analysis. So if I'll just go through what happens in Illumina sequencing workflow. So first you add adapters and along with the primer, and then you generate cluster. And for this cluster, there is a specialized flow cell, which have here it shows the 8 lanes, and each lane will represent one sample. And once your molecule is fixed in this flow cell, then there will be a synthesis by sequencing. When I say synthesis by sequencing, we in Illumina we use a specialized terminator containing nucleotides. So what happens once one nucleotide goes and bind to the complementary site of your fixed cDNA, then it will not allow the other base to come and start the whole chain of reaction. And since it's the fluorescent label, so you'll get to know which base has gone and complement to the your fixed cDNA, and that will give you a clue that which base is there, complementary base is there. And then again the second cycle starts, where again you will image the 4 colored bases, and then you will remove the terminator, and then the cycle will be keep going on till the full length of the cDNA. So this helps in, definitely it helps in fasten up your process, the whole sequencing process. Now coming to NGS data processing. So once you have the raw data, what you do after that? So there the bioinformatic analysis comes into picture. So if you have a raw data, you do data pre-processing. So what is data pre-processing? Basically, you trim out all the adapters, you remove if there is some biasness of an adapter towards any particular sequence. Generally, we use a kind of randomized adapter. So this is one of the major problem we see, because what happens once we have the final reads and we try to validate it by using qPCR or any other technique, Northern blot, we find that although we got a very high number of a particular, let's say, read, but when we are doing validation in the lab, we are not getting that sequence. So this can be because there is some biasness of this adapter towards a particular sequence, and that's making it multiple copies. So this is one challenge we generally face when we do go for Illumina small RNA sequencing. But as I said, this can be easily rectified by randomizing the adapter sequences. So once you have trim off all those adapter sequences, and then you have size selected, because for small RNA generally we take 50 base pair as in read size. So you remove others which are smaller or really big, and then you do a quality control in terms of the quality of the sequence. So there's something called Fred quality score. So it measures the quality of the identification of nucleobases generated by the automated DNA sequencing. So higher the quality score, that means that the probability that there will be there will be an incorrect base will be really low, right? So 30 plus at least, to see if there are RNA-seq data and mRNA-seq data, then we restrict it to 30. But if it is small RNA, I think it's always good to go for 30 plus. Now, once you have good data, good sequence with you, now the next step is to map them onto the genome. So working with, let's say, infected sample. So you should take both the genome. So in, let's say, in a scenario wherein you're interested in finding out the differentially expressed small RNA in a viral infected sample. So you will take the genome of the host as well as the virus and try to map all the reads onto them. So this is this step I was talking about. So this is, and also you should include the small RNA which are already annotated. So there are many databases. For microRNA, we have Mirase, and then there are RNA bases. You can use those and map them, and that can result into two different groups. One is the known small RNA, another one is which are not known before. So these are, we can put it in the novel small RNA category. And once you have these known as well as novel, then you can check for their difference in the expression level with what we call as DG, differential gene expression, or differential expression in the small RNA. And further, you can validate at this level by qPCR or Northern blot, or you can use as differential cell gene expression data to make some biomarker to define whether that particular sample is infected with virus or not, or you can go for a target prediction. So depending on what you want from this NGS, you can choose any of these three paths. This is just to give you a glimpse of the visualization of data. Once you have the data and you want to show the audience, or you have to publish the data, so you can use a box plot, you can use a welcome plot, you can use a heat map. This will give you a quick representation of whatever data you have got instead of going through each and every read. You can also go for this kind of donut pie chart. So this is just a representation, and it's always good to represent your data in a best logical and easiest way to be understood by each and everyone who just have a first glimpse. Now, here where I really wanted to spend some time, and that is proper planning. The first and the very crucial step for NGS is proper planning, as I reiterating the same thing. And what I mean by proper planning, so the proper planning actually is basically a decision making. So and the four important factor one should consider when you are going for NGS. First one is why you are going for NGS. What are the outcome you're expecting? So for example, in small RNA, the two most popular reason why people go for NGS is, number one, we want to do a differential expression profiling of small RNA in, let's say, disease versus a control sample. Or we just wanted to know the small RNA profile. That means we wanted to know whether and what all small RNA are present in a particular sample, right? So this two example I will be taking. I mean, all the time I will be taking these two examples to explain you other factors and parameters. Now, when we know that okay, we are going for this reason, let's say either for profiling or either for differential expression. Now the second question is how much depth or coverage we want. Well, frankly, when we talk about depth and coverage, it doesn't completely, I mean, what called validated. Well, it's not completely valid on small RNA sequencing. Is generally we use this term mainly for mRNA sequencing or genome sequencing. And so what is coverage or depth of a sequencing? It's the number of unique reads that include a given nucleotide in the reconstructed sequence. So, and it calculated as the number of reads you want, multiplied by what is the read size, read length one can expect from that particular platform. and divided by the assembly size. The assembly size can be anything. If you are doing for a particular gene, it can be a gene size, or if it is a chromosome, if it is a genome, that can be a genome size. So you calculate this and you get to know what is your coverage. Now, what are the factors which affect the coverage, or the coverage? What are the factors which the sequencing coverage depends on? Number one, the read length. As we can say, the read length, more the read length, more is the coverage. Genome size. So bigger the genome size, you require more read, right? Because it's inversely proportional. Application, definitely, as I mentioned you before, why you are going for the sequencing. Because if you are only want to know that whether the particular small RNA is differentially expressed in one tissue versus the other tissue, then I will recommend, instead of going for higher coverage, you should go for multiple biological repeats. So what are these multiple biological repeats? That I'll let you know. Then comes the gene expression level. So when we talk about gene expression level, definitely it means that if you really want to find out any meagerly expressed gene, or the gene which expressed in very low amount, low concentration, in that case, definitely you need a very high coverage, right? So if you really want to find out any lowly or meagerly expressed gene, you have to really increase the coverage of your sequencing. Genome complexity and repeated region, definitely you need more coverage, because you want to cover all these repeated region, and generally the polymerase and all those play a role. There are chances for slippage. This coverage also depends upon which machine you are using. So if you know that the rate of error is high, it's better to go for more coverage. And finally, the assembly algorithm, because the algorithms are depending on, our assemblers have a different requirement. So so depending on which assembler you are using, you can select your coverage. Now, as I already mentioned, sometimes it's not the coverage or depth which will give you significant read, or sensible or logical read. It's the number of biological replicate you are using. So for example, as I mentioned you before, that if you really want a differential expression, small RNA differential expression profile, it's better to go for multiple biological repeats instead of going for a deep sequencing of a particular sample. You before that this coverage and all parameters generally don't fit very well with the small RNA analysis, because for a small RNA analysis, just to do a small RNA profiling, I think 1 to 5 million read per sample is good enough. But as I mentioned you, again reiterating that if it is about comparison between two samples, it's always better to go for a more biological replicates. This is just give you a kind of glimpse of what all should be a number of read per sample if you are going for a particular experiment. Like if you have to do a gene expression profiling, your number of read should be in the range of 5 to 25 million reads per sample. For just a global view of gene expression, 30 to 60 million reads. In-depth view of transcriptome or assembly a new transcript, this is like 100 to 200 million reads. But as I said, whenever there is a comparison of expression, it's better to go for a multiple sample than going for a depth in the particular sample, right? So, and for the targeted RNA expression, the recommended is around 3 million reads per sample, and small RNA, as I already mentioned. Now, after the depth, we'll talk about the proper sample size. So more often, I have seen students and researchers giving one sample or two samples, and they expect that we will find some differential express small RNA or gene. It's absolutely a very wrong practice. See, I understand that there are chances that the sample is rare, right? But in that scenario, if the sample is rare and you cannot get more than 1 or 2 sample, in that case, it's recommended that you go for more read production. But again, after you get the read, that is not statistically significant because you have not given enough input for doing any statistical analysis. So my another recommendation is, once you get the reads, once you get the data, please do multiple validation technique. Please use multiple validation techniques. You have to validate that data, because depending on only 1 or 2 samples, you cannot conclude about the expression of that particular gene. So if you have a rare data, if you have a rare sample and still you want to go for NGS, you have to do a cross validation once you get the result. But and if it is not rare, I recommend at least 5+ samples. Biological replicate should be given, especially for differential expression analysis. Even 3, I will say it's not enough. Yeah, because what happens with any chance, if you find that one is behaving absolutely as an outlier, you cannot compare other 2, right? So it's always good, at least you should give 5 samples for any kind of differential expression analysis. Well, people also get confused about replicates, and I have been mentioning the biological replicates and technical replicate. So it's just a picture to show that when I say biological replicate, it should be from 2 different individuals, right? Whereas a technical replicate means the individual is same, but the library has been formed twice from the same sample. In some cases, we also do a sample pooling when either we don't have too much of money or the sample is rare. In that case, you pool these 2 samples, but once you get the data, then you have to individually check in these 2 biological samples that which read is belong to which sample, right? So yeah, that's all. So these are some of the important factor I wanted to talk about. And I think there is one more factor which was written statistically significant. Yeah, so this is my suggestion. Always accept any read when you find that read or the expression of that particular read is statistically significant, right? Otherwise, you end up with a wrong interpretation. Right. So this slide is just to show that, and it's a very common scenarios that we have a lot of reads and we generate reads because we have money. Some new projects have come, but we don't know what to do with those reads. And sometimes, the researchers and the students are at the 5th year of PhD and they still don't know what they have to do with these reads. So I just hope that you should not stuck in this whirlpool. So plan well. Now, coming to my research and how I use NGS as a tool to address some of my research question. My interest is to understand the role of small non-coding RNA mediated defense and counter defense in the antiviral immune system of silkworm. Silkworm, as we all know, is a producer of silk, and it gets infected with a double-stranded virus, which is a baculovirus called as a Bombyx mori nucleopolyhedrosis virus. This virus is a deadly virus, and it causes more than 80% of mortality every year. And since India being the largest consumer of silk, it's a big concern in sericulture. And because of this loss, every year we have to import thousands of metric tons of raw silk from China, and that is actually affecting our economy, and especially the present scenario of Corona. So to solve or to address some of these main issues, I have set some of my research interests. So first and foremost important thing is, I want to know if RNAi play any role in this host virus interaction. So I wanted to identify all the important component of RNAi and want to check whether there is a crosstalk between siRNA and microRNA biogenesis pathways. Second, if there is, then what are their targets, and whether only host is generating small RNA or virus can also generate microRNA, siRNA, or piRNA. And once we have a clue about that microRNA or siRNA or piRNA playing a role in host-virus interaction, I wanted to do a kind of a microRNA profiling, which can be used as a marker to define resistivity and susceptibility of any host against the virus infection. And finally, I want to develop a virus-resistant insect strain using synthetic microRNA against the essential genes of the viruses. So this is what I generally do. So I have different projects. So I deal with both long non-coding RNA and small RNA and also mRNA. But for today's talk, I will only concentrate on the small RNA and the project what I am handling and what we have already used. So I have selected this particular project wherein our NGS helped us in discovering 5 microRNA encoded by virus. It has been published and cited by multiple research groups, and it also recommended in the faculty 1000 as the best read for that year. So this was all because because we did a really good I'll say decision making before setting up the whole experiment and subjected into NGS. So what we did, we isolated the tissues from the host after infecting it by injecting the virus molecules at 12, 24, 36, 48, and 72 hours. And we isolated the RNA from these infected tissues. And we also use the respective control for all these different time points. And for each time point, I have sent 3 samples as a biological replicate. But actually, I have 5 samples just because I didn't have much money at that moment. So I gave 3 samples each from these different time points. And so this is again the same. So what is the whole methodology we used for the small RNA prediction? So you can see here, isolation of RNA from fat body. Fat body is a kind of a liver in human because it is kind of a powerhouse of all the immune genes. So we were speculating that if it is something related with a viral infection, definitely it should have something to do with this tissue. And then size fractionation. So we wanted small RNA less than 30 and more than 17 nucleotide. We subjected to so next generation sequencing. We did a Illumina sequencing, HiSeq 2500. And then we mapped these small RNA onto virus as well as the whole genome. And then we did a screening. And finally, we did the experiment validation. So now I will talk about from here. So because these are all technical things which can be easily taken care by any good platform service provider. So once I got the small read and I mapped them onto the virus because I was interested in the small RNA encoded by the virus. So I took a cutoff around 18 nucleotide or more, an e-value of 0.1, and I found there were 24 hits which were mapping on the viral genome. Then I also blast them these 24 onto the host and try to find out if there is some overlap. And once I did a blast, I found that 3 of them were overlapping also onto the host genome. So I omitted them. And then I checked for, because they are small RNA, it may happen because of the host immune response. They could be the part of some viral ESTs. So I remove that those fragments also. And finally, I've got 10 unique hits which were mapping only on the viral genome. And then out of these 10 hits, I try to find out if how many of them can make a precursor structure. And then I found there were 5 small RNA, which I can with confidence call them as a microRNA. And then when I tested them using Northern blot, 4 of these 5 were also shown expression in different tissues. This is fat body.
Yeah, the session will be starting in another 2 minutes. There was some network issue on from the other end. Just wait for 2 minutes, guys. Ma'am will be rejoining the session in another 2 minutes. There was some issue with the network. Ma'am will be joining in another 2 minutes. Guys, till then you can utilize this time in dropping your queries in the chat section. If you have any queries regarding the topics that were covered till now by Dr. Jyoti, you can drop in your queries in the chat section. I'll be covering them once ma'am is done with her talk. And
Hello, am I audible now?
Yes, ma'am. Yes, ma'am. You are.
Sorry for the interruption. The light went off. Okay, can I start again?
Yeah, sure, ma'am. Sure,
Yeah.
sure.
Okay, so we also found that four of these five microRNA were expressing in two different tissues. So it's a good sensitivity, because the fifth one we could not find, and then that's what we published. The next thing what we did was we were interested in finding out the targets of these viral microRNA. So we grouped them into two groups. First one, it was the viral microRNA targeting their own genes, which we call as autoregulation. The other one was the trans targets, or we call it as the protection against host. And we found that this virus utilizes its microRNA to combat host antiviral response, because we found almost all the innate immune response genes were the target of these viral microRNA. We published this data again, and I will talk about one of that viral microRNA, which was found to suppress RAN. RAN, as we know, in the microRNA biogenesis pathway, RAN is the cofactor of exportin 5, and exportin 5 helps the precursor microRNA to get transported from nucleus to the cytoplasm. So I'll just show you the final model what we gave. I know it's a messy slide, but I'll try to explain you. So what this microRNA does. So when there is no viral infection, the cell is happy, and the host is making, this is a host microRNA, you can see the host is making its microRNA with the help of DROSHA, and then exportin 5 is exporting these microRNA from nucleus to the cytoplasm, and then these host microRNA can act against any viral invasion. And you can also see the RAN is getting the RAN mRNA is getting transcribed. But what happens when this virus produces this particular microRNA? This when the virus enters the cell, the host cell, it starts preparing its own microRNA at the initial time point, let's say before 24 hours, and it produces this microRNA. And after 24 hours, what it does, this microRNA goes and blocks the RAN mRNA, the host RAN mRNA. The viral microRNA is blocking the host RAN mRNA. And by doing that, it will stop the transport of the host precursor microRNA from the nucleus to the cytoplasm. And if there is a decrease in the transport of precursor microRNA from the nucleus to the cytoplasm, there won't be a host microRNA available to combat the virus genome. And that's how the virus will overpower the whole host machinery. So this was one model we came up with. The another model was, as we saw, that the viral microRNA is also targeting its own gene. So the question arises here is, why virus has to produce microRNA against its own gene? To do that, we selected this particular viral microRNA, which was targeting all these viral genes. And the interesting part was that particular microRNA expresses in the early stage of infection, whereas all its target genes are they express at the late infection stage. When I say early and late, so this particular DNA virus has basically two phases. One is the early phase, wherein the virus goes inside the midgut and start establishing its infection. And once it's done with establishing all its machinery important for the virus multiplication, it now the next stage, which we call as the late stage, it started a kind of a horizontal tissues, and then it produces its protein and then comes out from the host. So now this was interesting, that the microRNA which was targeting the viral genes was expressing in the early stage, whereas all these targets were expressing in the late. So now the question was, why a viral microRNA has which is expressing in the early stage has to suppress the genes which are expressing only in the late stage. So we came up with this model. So what it states that if this particular microRNA, which is expressing in the early stage, does not block the mRNA, which are expressing in the late stage. So if it is expressing in the late stage, it's quite obvious that their role is only in the late stage. So they don't have any role in the early stage. But what happens if the virus will produce this mRNA in early stage? The host will get this clue because the virus, the load will be very high, and then the host will get the signal that look, there is a viral invasion, and now we have to prepare and we have to fight back. Not to hide this kind of signal from virus to the host, they suppress all these late gene in the early stage. So you can see over here, this this virus microRNA expresses in the early stage, and then it goes and binds to the late expressing gene in the early stage, and then that's how it escapes the recognition of the host defense mechanism. But in the later stage, there is no expression of these particular microRNA. Then these late mRNA are free to express, and but then by that time the virus has already overpowered the whole host machinery, so the host cannot do anything. So these were two important strategy we came up with. The other important thing, you remember this slide wherein I showed that 10 small coding RNA, 5 were microRNA. So we were also interested in what are these other 5. So when we checked this 5 and their expression, we found I'm just showing one of the data of these siRNA. So what we found that this this small RNA was indeed expressing, and to find out which pathway it is following, we selected DRYSER2 and ROSHA. DRYSER2 participates in siRNA biogenesis pathway, whereas ROSHA participates in microRNA biogenesis pathway. So when we knock down DRYSER2, we found there is a decrease in the expression of this particular small RNA, indicating that it follows a siRNA pathway. Whereas there was no change in the expression of this small RNA when we knock down the DROSHA. Again, reconfirming that it is not following the microRNA pathway, it is following the siRNA pathway. Now we are still in the process of finding out the target of this particular siRNA. But to tell you the truth, this will be the first report where we are showing a DNA virus is encoding an siRNA. We also do a kind of small RNA profiling, which is the present work I'm doing with the help of BioInnovate, wherein I'm interested in understanding the role of differentially expressed microRNA between resistant and the susceptible strain. And for each of these sample, I have given 333 biological replicates, and we are still in the process of data analysis. So I think that's all, and I will end my slide again on this very important point, which is decision making. And I don't want to see anyone stuck up with NGS garbage, and so plan well. And now I think I'm ready to take up questions. Thank you for your patience.
Thank you so much, ma'am. There are a few questions that have dropped in.
Okay.
Just a second. So first, like there's a string of questions posted by Mr. Dipankar Mandal. He starts his queries with like the first question that he has asked is related to the adapter sequences. He's asking, are these adapter sequences universal, the ones which we use?
The question, yeah. So the answer is yes. For these adapter sequences are generally universal.
Okay. His next question is, uh, 1 second. His next question is, after whole cell RNA isolation, how is the small RNA enriched or differentiated from other RNAs like mRNA, etc.? Or do we send all RNAs for NGS and later differentiate?
Yeah, so we generally send the whole RNA, and then with the size fractionation, we cut off only the size which we are interested in.
Then he's asking, ma'am, were your samples infected fat bodies of silkworm in which virus has integrated its genome?
Bombyx mori nucleopolyhydrosis virus, because it is very specific to Bombyx mori, and it has been co-evolved with it. So there are high chances that the virus or the host genome must have been integrated with each other.
Okay, then Miss Pranita is asking, she's asking for suggestions pertaining to some bioinformatics analysis of already available data that she can do during this lockdown period. As we lack funds, we are an academic institute.
What particular things she wants to say?
Like she's asking available data sources. I think she can try for SRA. She can download the data from SRA and
Yes,
start practicing.
yeah. So depending on whether she's interested in only microarray or she is interested in all kind of. So
Right. There were a few questions regarding COVID-19 as well.
okay.
So, if shall I bring them up, ma'am?
Yeah, I mean it will be interesting. I wanted to see like, what kind of question regarding COVID.
Okay, so they were asking, one is why we go for PCR test to confirm COVID-19, whether the person is positive or not.
Good.
So why do we go for PCR test to confirm that?
So first question. So I'll answer that. So PCR is very sensitive technology, and when we say that a particular person is infected with virus, and you try to take out the sample, whether it's a blood sample or from the mouth, from the nasal cells, you will find a very less amount of viral particle in that. Okay, so if you have a very small amount of viral particle, you need a amplification of that particle, right? And that's why we use even if the virus is in a very very small amount, using PCR can easily detect its presence in that particular sample. So it's a very sensitive technology. Even virus in a very less load, we can easily identify the virus. So that's why we go for PCR.
Okay, and the next question is the role RNA sequencing is playing in vaccine designing as far as COVID-19 is concerned.
For vaccine designing, to my limited questions, limited knowledge, see, vaccine designing definitely, because see, RNA-seq is very powerful technology in terms of sequencing the whole viral genome, this particular viral genome, right? So, and it's a RNA virus, right? So definitely when we make some attenuated virus also, or anything which is somehow matching with a kind of a chimeric segments, we need the sequence information. And that sequence information we will only get if we have a genome sequence of that particular virus. So I think directly it is playing a role, because if we don't know the sequence, it will be difficult for us to make any chimeric or any kind of what do you call that, attenuated virus. So that's how it is playing a major role in the vaccine development.
Okay, and there was a question about flow cell. What is a flow cell?
Okay, flow cell. Do you want to answer this, Avinash, or because
yeah,
yeah.
If you could start, I mean, I
So
can.
okay, so
Yeah.
flow cell it's a small what do you call it, template, right? And it has different lanes, and in the flow cells it's a kind of thing, you need a molecule to adhere somewhere, right, before it will start cDNA synthesis. So flow cell is a kind of that matrix, wherein your RNA goes and adhere, right?
Yeah,
And
right.
then all the clustering and reverse transcription takes place at that particular matrix. So that's what I think I know.
Yeah. And Mr. Shubham is asking how we can manage the coverage and depth for the experiment.
Manage. So as I told you, it depends on what you want. Manage means, see, the machine will take care of it. So if you want more read, you tell the service provider that you want this many reads. So based on your demand, they will decide on which platform you have to select. So, and why I have told you already that if you really want any new or low expressed low expressed mRNA to be, to study, then you have to go for really I mean, high depth. But for small RNA and all, I don't think depth play a very crucial role, because the sizes are very small. And if you will calculate with that formula, you will realize that you don't need that much depth for sequencing of the small particular RNA.
And moreover, if we talk about the coverage part, it is almost uh, proportional to the number of cycles as in for which the machine is being run. So if you want a higher coverage, you can go ahead and increase the number of cycles for which your machine is being run.
Okay, he wants to ask how to generate.
I think it was towards that tangent only. I feel.
Okay, so then that means you have to really increase the cycle. Okay, if
Yeah, right.
the question that means how can we increase the coverage? Okay, yeah. So
Yeah.
yeah,
And then another question is about what precautions you take while working with small RNA samples, as RNA are very unstable
what precaution we take? Okay.
while working with the samples.
Yes, there are a lot of precautions you have to take. First of all, you should never let the RNA be at room temperature. So maintain the temperature. It should be. You should always work keeping the samples on ice. Number one. Then storage is another very important aspect. So you should always store your RNA in -20 if you have to use it, and if you're working on that particular RNA continuously. If not, it's better to store at -80. And you should always use autoclave gloves, autoclave pipette tips, and try to not talk while you're isolating the RNA. So, and then whatever you are using, it should be autoclave or it should be new, sterile, and completely nucleus-free water you should use for adding it and for diluting it. So that is important. So these are the few of the precautions you have to take before isolating the RNA.
And Dr. DK Sharma is asking about your views on 16S RNA as a medium, 16S RNA sequencing as a medium for bacterial identification. What are the limitations of this technique?
So, frankly, I did not work on the bacterial metagenomics, but I think this is the most powerful one, because that's how, because there is a lot of variation in 16S rRNA, and every group of bacteria has a specific telltale feature about 16S. So that's why 16X is a very powerful what do you call tool to what do you call to classify these
Yeah, right. Metagenomics, what we call as metagenomics studies, 16S RNA is like primarily considered
correct.
to kind of
So,
differentiate, because see, we are limited by our bacterial culturing technique. So this is a very powerful tool in terms of classifying and identifying
yeah.
the bacterial samples that are present. And there's another question why, regarding your work, ma'am, why did you choose miRNA and not any other RNA for Bombyx mori infection in SIL? There can be other RNA playing some role in the same process. So what was the reason behind choosing miRNA and not any other RNA?
So the study, what I showed was viral microRNA, and since this is a DNA virus, right? So in case of DNA virus, the chances that it will make siRNA is low, right? Because the double stranded RNA availability in DNA virus life cycle is rare, right? So generally DNA viruses produces microRNA, but as I said, we also found some of these siRNA. So it happens when you know that one particular type of virus produces one particular molecule, so you first start your research with finding out what is known. So DNA viruses were only known for producing microRNA. So we first checked for microRNA, but we did not limit we did not limit our search only for miRNA. We also found siRNA. That's what I showed, that we also found there are small RNA which were not following the microRNA biogenesis pathway, which were following the siRNA biogenesis pathway. And as I mentioned you that we started with 24 viral microRNA and then we left with 10. And in that 10, we grouped them into microRNA, and the second group was small RNA. So that can be siRNA, that can be piRNA. We are still in the process to find out which group it belongs to. So it was not intentional. It was just to start with, we selected microRNA.
Okay. Can you suggest some from which we can get list of is there any online database? Another question, ma'am. Sorry. Can you suggest some online databases from which we can get the list of small RNAs in cell?
In cell?
Yeah.
For microRNA, Mirbase is there, right? Then there is a small RNA database. You can use that, right? That they both are publicly available for downloading. So these are the two which I generally use. See, the thing is you need a curated data, right? You cannot use any database, because sometimes what happens, people will just do an NGS of small RNA and then they will upload the data elsewhere, right? So if there is no good curation of that data, I will not recommend them to use them. So I think Mirbase and small RNA database is two of these databases which you can use. Hello?
Hello. Yeah. In addition to what ma'am said, just adding on, there is another database called RNA Center, which can also be used in this scenario.
Oh yeah, sorry. Yeah.
Okay.
Correct.
So, just a second now. There is one more question. A lot of questions coming up. After mapping the sequences, again Deepankar, after mapping the sequences to genome, do you perform validatory experiments? Can you please highlight how did you narrow down from 24 small RNA sequences to 10 small RNA sequences after mapping?
After mapping, yeah. So because I was only interested in the viral small RNA, so if it is mapping onto host also, right? So I thought there is a high chances that because what happens, the sizes are very small, right? So the chances that it will have some kind of complementarity or some kind of complementarity with your genome is very, very high. By chance, what I'm saying is by chance. So you get a lot of false positive, right? To omit that one, I again mapped those 24 sequences on host. I found the matching extent or the mapped bases were more in host than the virus, because as I said, they were not the complete matches they were not matching or completely on the viral genome. The match level was more than 18 nucleotide, and the energy I took was the e-value I took was more than 0.01. So if it is exact match from the virus, I have taken it. But if it is mapping more on the host as compared to virus, I omitted them. So that was first criteria, that if it is matching more on the host, I just thought it may be a part of host which by chance is mapping onto the virus. So that was first criteria. I did not validate any of the data at this level. I further went and checked for the microRNA and siRNA. So that was the reason why it went from 24 to 10. And another thing, I think not only mapping, there was another thing which I has used was EST, viral EST. So what happens? See, because when you isolate the tissue, the cells will not be in a sync, in the sense that in some cells you will find that the host is fighting against the virus, and there is a degradation of the viral EST. So those small RNA will also come when you do the sequence. So there is a possibility that after degradation of that viral EST, the size of the degraded product is somehow similar to the microRNA size product. So to omit that possibility, what you do, you check for multiple samples, right? That's all. So in all the 3 multiple samples, you will find the same sequence. Then you can say with surety, okay, this is not by chance, this is not a degradation product. Because the degradation of any kind of EST will not always produce the same degraded product. So this is one way to check whether the small RNA is actually a small RNA or it's a degradation product of any EST or any mRNA. So that was another criteria I used. So for 24, I came down to 21 by omitting all the host small RNA. And then from 21, I came down to 10 because I found that most of them was the degraded product of the EST.
Okay. Ms. Divya is asking, is the mechanism for the generation of microRNA from DNA virus and RNA viruses, are the mechanisms same for both the viruses?
RNA viruses generally don't produce microRNA as per my knowledge, right? But see, method is little different in terms of where they're getting produced. The DNA virus goes inside the nucleus. So there are high chances that it will produce a precursor microRNA. In case of RNA viruses, they don't go inside the nucleus. They multiply inside the cytoplasm. So the chances that they will make a precursor microRNA is low. So this is a major difference in when we talk about the microRNA from DNA virus and the microRNA from RNA. We will not call them as a microRNA if in any situation any RNA virus is known to produce microRNA. We can call them some other name because they don't form any precursor microRNA. We can call them, let's say, intronic microRNA or some other name, but not the typical microRNA. They can be some other small RNA. They cannot be microRNA.
Fine. Final question. Is this DNA virus which infects Bombyx mori, is it always beneficial? I mean, do they have some kind of symbiotic relationship, or it has some adverse effects on the host as well?
There is no symbiotic relationship. This is a lytic virus. It always kills.
Okay.
Although they have co-evolved, so you will find many of the integrated sequences in host as well as virus, but there is no symbiotic association it has with the virus.
Okay. I think that is pretty much it. I think we are done with all the questions. Thank you so much, ma'am. Thank you so much for sharing your time and knowledge with all the participants, taking time out of your busy schedule and interacting with the audience. Thanks a lot, ma'am. Thank you so much.
Thank you, and you all are most welcome.
Thank you. Thank you to all the participants as well for patiently listening and sitting to the session, sharing their queries and interacting with ma'am. Thank you so much. I would like to end the session. Okay. On behalf of Bionovid Technologies Private Limited, I would like to thank Dr. Jyoti Singh for sharing her time and all the participants as well. Thank you. Thank you, guys.
Thank you, Avinash.
Thank you.